This Week in Virology, the podcast about viruses, the kind that make you sick.
From Microbe TV, this is TWIV, This Week in Virology, episode 810, recorded on September 28th, 2021. I'm Vincent Draccaniello, and you're listening to the podcast all about viruses. Joining me today from Ann Arbor, Kathy Spindler.
Hi, everybody. Here it's 68 Fahrenheit, 21 Celsius, perfectly blue sky, not a single cloud. Really nice day.
20C is just finished raining here. I have a very different view now. This is the incubator. And I look up at tall buildings. I can't see the street. I see the back of a billboard here. If I walk to the window, I can see 7th Avenue and all kinds of billboards. Right up the street, there's a big billboard of, what's the guy that plays James Bond?
Daniel Craig?
Daniel Craig. Like 20 stories high picture of him. When I come out the front, I see him. Why don't they have a virus there instead? Also joining me from Austin, Texas, Rich Condit.
Hi, everybody. 92 degrees, a few clouds, sunny. It's okay. I keep saying that we're on our way out of summer, and that's kind of sort of true. If I look at the next, I don't know, what is it? Over 10 days forecast for whatever that's worth. There are no other days upcoming that are 90 degrees or more. So that's good. McFadden told me the other day that it's cooling off in Phoenix as well. So that's a control. So, I think we're on our way towards weather that is less hot. We also have, I can hardly believe this, but the caseload in Austin is plummeting.
Good.
And we're down to, they've officially declared stage 4 today, but in terms of the actual 7-day moving average of hospitalizations, we're well on our way to stage 3. Okay. And this is going to, the pattern of these surges is going to keep epidemiologists busy for decades, I think. Okay. We could discuss that another time, but that's just part of the weather report.
What has vaccination gone up? Do you know?
Not. I don't think sufficiently to account for this. Some, yeah, and there's a good news, bad news situation where the schools have started. That's potentially bad news, but in defiance of the governor's mandate, a fair fraction of them have mandated masks. And there's new data out you've probably seen for these and other school data that show a correlation between whether or not there's a mask mandate and the caseload in a school. So masking works. Masking mandates, not just masking mandates appear to have an effect. Now, the thing that strikes me is that there's been no official restaurant shutdown or bar shutdown or anything like that, as has happened previously when cases decreased. The only thing that's happened is that locally, Austin the public health department has put together guidelines that basically in stage 4 and stage 5 say that you ought to be wearing a mask if you're in a group, either indoors or outdoors, whether or not you're vaccinated. And I noticed that from just anecdotally, me being out and about, that there was, I think, a very significant increase in mask use from where this particular surge started up and between then and now as it's resolving. Okay. So, let's just say I want to believe that this is mostly a behavior thing. Okay. And if that's the case, then I think that the resolution of this particular surge may be a strong argument for the efficacy of masks in the general population, just on a day-to-day basis, because they've not closed the restaurants, they've not closed the bars, they've not closed the parks, they've not done any of that. The only real difference that I can see is a strong recommendation locally to wear masks that I think is being adhered to a significant extent, not 100%, but I think significantly. So it's all very interesting, but it's good news.
Good. Also joining us from Madison, New Jersey, Brianne Barker.
Hi, it's great to be here. It's 66 Fahrenheit. It is not currently raining, but it's been on and off all day. And I think I have all of the clouds from Rich and Kathy's skies that allow them to have cloudless skies. I think they're all here.
Did you have rain this morning? We did.
We've had rain on and off all day. I was outside about an hour ago, maybe 2 hours ago, and it was raining still then.
So here in New York, we have vaccine mandates for restaurants, healthcare workers, many other people. I went to lunch the other day with a former student here, and they asked to see my vaccine card. I'm like, oh, fortunately, I took a picture of it months ago, right?
And that was good enough.
That was good enough, yeah. They weren't going to send us out. They sat us and they said, oh, take your time.
I noticed New York is taking a really hard line with this.
They are.
And that's very interesting. It reminds me of when the air traffic controllers went on strike during the Reagan administration.
Yeah, right.
And he said, okay, fine, you're fired.
You fired them all, yeah. A lot of healthcare workers don't want to get vaccinated.
Yeah, I don't get it.
Yeah, thousands, apparently. It's crazy, yeah. But on the streets, not everyone wears masks. 50%, I would say. But in the mass transit, you have to, in subway, trains, etc., restaurants. So, yeah.
Do you find good compliance in places like that, like in the mass transit?
Yeah. So on the commuter train, New Jersey Transit, which they tell you to get off if you don't have a mask. And everybody wears a mask. It's interesting. On the subway, there's no policing. There are no police on the cars pretty much. Yet, pretty much everyone wears a mask, and properly, right, over the nose and so forth. So it's actually quite impressive.
Well, that's
people
good.
Get it here, it seems to be. At least here downstate. I'm sure if you went upstate, it would be very different.
Probably. I flew this past weekend. It was the first time I'd been on an airplane since March of 2020. And I was not always impressed by which parts of the face people thought needed to be covered with their mask.
Yeah, I don't get it. I don't understand people who, it's obvious. And I think people are just being weird, almost defiant. I mean, walking around with a mask on your chin makes no sense. I mean, why bother at all? I mean, because that's not doing any good. It's more like it's kind of thumbing your nose at the whole thing or something like that. I don't get it.
I drive to the train station. On the way, I pass school crossings, right? And they're mostly elementary. And it's interesting, the parents are walking the kids. And about half the kids have masks and half of them don't, right? It's not mandatory. But clearly some parents, even tiny kids, they're masking them up. It's interesting.
I think, at least around here, outdoors, people aren't compelled to wear masks.
Yeah.
Once they're inside, yeah. But so I could see where, some of those kids that don't have masks, they may put them on by the time they get to school.
Sure. Maybe the littler kids, they put them on at home just to get them. Otherwise, they'll run off when they get to school.
Yeah.
I was also wondering about this thing that, it's been publicized, at least here recently, about the correlation between, or shall we say, inverse correlation between mask mandates in schools and caseloads in the schools. The places with mask mandates have lower caseloads, but, there's a lot of potentially confounding variables there. Okay.
Yeah, of course.
I wonder whether communities that defy the mask mandate in general comprise a population that takes the whole thing more seriously. Okay. And that could impact on what you see in the schools. But generally, it does support the notion that behavior counts.
Yeah. Mm-hmm. So we have, as you know, Democratic governor in New Jersey, and he's running for election, and he has a Republican opponent whose platform is no masks, no vaccines.
Can you believe that's a political platform?
It's crazy.
It's nuts.
Playing with people's health, right? It's crazy. Unbelievable. But there are people who respond to that. Mm-hmm. Totally. In a way, you know, I don't like this pandemic, but it's really interesting to go through one to see all the stuff that happens. Because I don't know about you guys, I don't remember any of this during the 2009 H1N1. Do you?
No.
That blew through so fast. It was over before I hardly knew what was going on. Yeah. You know, I think that to me was a different kind of phenomenon. It didn't linger. It didn't kill to as many people. I don't know about the case fatality rate, but it certainly didn't. It didn't plug up the hospitals the same way as this. So okay. Yeah. Zika blew through the population really fast as well. And in terms of the consequences, that was pretty selective, you know, pregnant women, and in a certain geography, really, where it was circulating. But that also blew through really fast, which, you know, this lingers for reasons I don't fully understand.
The next flu pandemic, it'll be really interesting to see how many people mask, because in 2009, nobody masked as far as I could tell. But I wonder if the next one, they'll say, hey, this really helped. I would. Yeah. Right? It makes sense to me, because I think that, you know, in aggregate, all the data suggest that it really makes a difference. And other cultures, other countries have known this for a long time.
Yeah, I can see certain situations where I think I'll be wearing a mask for a very long time. Like when I'm on airplanes, it just seems to make sense.
So they didn't tell you to wear any particular kind of mask, just a mask?
They did not tell us any particular kind of mask. Actually, I think they said no vents in the mask and no bandanas.
Okay.
I know some international flights are pretty stringent about the kinds of masks that you can wear.
Yeah, I've heard that too. Yeah. Okay. Such are our reminiscences for 10 minutes here. We have a snippet in a paper for you. The snippet, something we mentioned on Friday briefly, but I wanted to, it's a very nice paper in Cell. It's kind of a review. As you know, the Lasker DeBakey Clinical Research Award was given to Catalin Carrico and Drew Weissman. And someone mentioned it Friday on Twiv very quickly. Maybe it was Dixon. And so I saw this little review article, and it's really nice. I thought we could chat about it because, you know, their careers are interesting. The whole story is interesting, and the science is really interesting. And this is a situation where mRNA vaccines have clearly had an impact on this pandemic, right? They were made very quickly, and that's because people were working on them for a while, as you will see. So this mRNA platform is here to stay. And so these two people played a key role. They're not the only people, but they have played a key role. Catalin Carrico, born in Hungary and got her PhD in biochemistry there. Moved to the U.S. in 1985. She was a postdoc at Temple University in Philadelphia, and that's where she got interested in mRNA, therapeutic uses of mRNA. Then she became a research assistant professor at University of Pennsylvania School of Medicine, and again working on in vitro synthesized mRNAs which encode proteins, of course, that would be useful for treating diseases. But interestingly, or not so interestingly, their work didn't go so well and didn't get funded, and was demoted to a senior research investigator. Yet she persisted and kept working and encountered, they write, the game changer was a fortuitous encounter with a newly arrived immunologist, Drew Weissman.
Supposedly that was at a copy machine.
Right. Which is not happening in the past year and a half, right?
Well, how often do people even do photocopying anymore?
Oh, copy. I thought you said coffee. Yeah.
Copy machine.
Yeah. I don't know. We used to have a big departmental copy machine in the hallway. We always used to run into people. Yeah. Chat with them. Anyway, Weissman came to UPenn in 1997. He had been a fellow at NIAID, National Institute of Allergy and Infectious Diseases, under Tony Fauci. Dun, dun, dun. How about that? It's a small world. And it turns out he and Carrico had common interest in therapeutic mRNA. Now, the problem was that mRNA is immunogenic, turns on interferon pathways, and make the cells that sense it make pro-inflammatory cytokines, particularly dendritic cells. So they had to get around that because that wasn't a useful property. But working on let me fix my scrolling is jumping here. I don't like it to jump. I need to scroll smoothly between pages. Thank you very much.
One might ask why it is that RNA is immunogenic. And if it is, then why aren't we totally inflamed all the time because we make RNA? And my answer to that, you can comment on this if you want, but I think it's relevant to this whole discussion, is that cells have ways of disguising their own RNA in a fashion that distinguishes it from foreign RNA, like you might expect from a virus, so that a cell can say, oh, that RNA is not from here, and mount an immune response. So the trick becomes, how do you make RNA look like more like cellular RNA?
Yes. And of course, the reason that we do have a response to RNA is because that's a really good target for detecting viral infection.
So it's quite ancient,
yes,
that detection of RNA, because viruses have been around a long time. As you'll see today, there is a particular way of disguising RNAs that they use. So they found early on that if you use small interfering RNAs to try and modify virus infection, they were looking at herpes simplex virus. They activated toll-like receptor 3, and that compromised the therapeutic usefulness of the small interfering RNA. But here's the key, and I love this is just something that just blows you away. They were using tRNA as a control in their experiments, and it didn't turn on innate responses. tRNAs are they have a lot of modified nucleosides. As I hear, up to 25% in any tRNA are modified. You know, I have to say, this is key. I used to say, who cares? Years ago.
To me, I was just going to say, this is like a eureka moment.
Yeah.
You know, it's, we all know that tRNAs have modifications. And like you're saying, Vincent, it's kind of like, it's just a property of tRNAs. But then they figured out this is really important.
For the newbies out there, I want to just say a word about tRNA. The T is for transfer. Transfer RNA is the adapter molecule in protein synthesis. And it's actually, well, I don't know, I suppose you could say the tRNA synthetases are the real key to translating a code. But, okay, they've got transfer RNAs are ubiquitous in cells, okay, because they're doing work all the time. They have an amino acid at one end that is appropriate for what's called an anticodon at the other end, which is a sequence of 3 nucleotides that base pairs with the appropriate codon in messenger RNA. So that's where the translation comes from, translation of the code, the nucleic acid code, triplet code into an amino acid sequence. So cells are loaded with these things. And we've known for decades that their tRNAs are loaded with modified nucleotides, all kinds of different modifications. So it's made by transcription with normal nucleotides, and then there's a whole slug of enzymes that come along and tinker with that and change the nucleotides to add things or rearrange things or one thing and another. And I think that impacts on structure, okay? There's been a lot of work put into this. Matter of fact, one of my very first projects was to look at modifications in ribosomal RNA and try and figure out what they were for. At any rate, but probably some of this has to do with immunogenicity as well.
Yeah. And when we say modifications, you can think to your chemistry, and if you think of ball and stick models, some of these modifications are just putting extra sticks or balls on various places.
Good point.
On the nucleotides. So a lot of them are modifying the U base, the uridine that we see in RNA.
Yes, I also have to say that Kathy mentioned this. She thought of this as a sort of a eureka moment. I sort of laughed when I was reading and saw how the authors of this piece dealt with that, because they talked about how this is where the penny dropped.
The penny dropped. Yeah.
Yes.
By the way, the authors are Lucy Shapiro from Stanford and Richard Losick from Harvard. And the name of the paper is Delivering the Message, How a Novel Technology Enabled the Rapid Development of Effective Vaccines. Yeah, I looked at that, how the penny dropped. Wow. They published a 2005 paper in Immunity where they said you can make RNA in vitro using T7 RNA polymerase. And that's another key technology here, using phage, DNA-dependent RNA polymerases to transcribe DNA templates to make mRNA. Without that, we wouldn't be able to do any of this, right? And we've talked about that before. There is a video over on YouTube of Rich cooking something in his kitchen in Florida, talking about the origins of T7 transcription. And that's key here. Anyway, they made this RNA with modified nucleotides. It reduces production of inflammatory cytokines from dendritic cells. Uridine derivatives, in particular, were very effective, like 5-methyluridine, 2-thiouridine, pseudouridine. As a student, I used to look at these. Oh God, what does that mean? Why is it there? No one would ever tell you what it meant. I'm not sure they knew. And you don't need to have a lot of modified nucleosides to have a suppressive effect. And so they published this paper in Immunity. It'd be actually fun to go back and do it sometime, right? The 2005 paper. They write at the end, these results are essential to the design of therapeutic RNAs. And they went on to show that pseudouridine reduced immunogenicity and increased translation of the mRNA in vitro and in mice. They say here that, and also, yes, not a lot of people paid attention to this, but a few people did. One person, Barry Poliski, he was the founder of Sirna Therapeutics, siRNA Therapeutics. And they made chemically modified RNA to increase its stability. And when they put these RNAs, these siRNAs encapsulated in lipids and put them in mice in a model of Hep B infection, these changes to the RNA lowered the cytotoxicity and the recognition. So they say here, the early publications garnered little attention, but were noticed by a former young faculty member at Harvard, Derek Rossi. He came across this paper and he used the approach to deliver the 4 mRNAs into human adult cells that you could reprogram into pluripotent stem cells, right? The Yamanaka factors that you can change any cell into a pluripotent stem cell. He used their technique to put mRNA in instead of plasmids. He founded a company. The company's name is Moderna. For modified RNA, founded in 2010 for developing mRNA therapeutics. They licensed patents from UPenn based on the Carrico and Weissman discoveries. He left Moderna in 2014 to do other things. I wonder if he's kicking himself that he left. Probably got a lot of money, right? I'm sure he has a lot of stock. In the meantime, BioNTech, founded in Germany in 2008 by Ugur Sahin and Özlem Türeci, they embraced nucleoside-modified mRNA. They licensed the patents to the technology, the Carrico-Weissman Penn patents as well. They wanted to make a cancer vaccine. Carrico ends up in 2015 being appointed a senior vice president in BioNTech and they decide to expand into vaccine development, while Moderna is exploring viral vaccines, mRNA vaccines like chikungunya, Zika virus, cytomegalovirus, and they push it forward by modifying these mRNAs with lipids, the modified bases, the pseudouridine, and they showed early on that you could, if you'd use an mRNA encoding erythropoietin, you could get really good synthesis 6 hours after injection of the mRNA. And if you don't modify it, you get 10 to 100-fold lower levels of the protein without that pseudouridine.
Rich has just put in a figure in our notes that shows that the difference between the uridine and the pseudouridine is really just a rotation around the molecule. So in this case, there aren't any more balls and sticks, as it were.
Yeah, I have an ancient fondness for pseudouridine because my Harry Noller, when I took biochemistry of macromolecules from him, he used to have a weekly quiz, and I don't remember any of it except one of the quizzes had a little thing about pseudouridine in it, and of course I got it like totally wrong, but it forced me to look at pseudouridine and see what it was, and I realized how really goofy this is. And I presume that the pseudouridine synthetase actually goes in there and just clips off the uridine and flips it around and sticks it back on. I don't think it's sticking on a new uridine, but I don't know that for sure. But at any rate, it's just an isomer, okay, where the uracil is stuck to the sugar using a carbon molecule that's in the ring rather than the normal nitrogen. Okay. And if you look at it, the base pairing properties are the same. Okay. It doesn't really it doesn't change the base pairing properties at all. Okay. It does change some of its properties, which was part of what Harry's quiz was about, but I forget. At any rate, yeah, I think pseudouridine is just an awesome molecule.
And Katie Carrico, in the lecture that I heard, I think that's where she said it that she doesn't technically consider this to be a modified base. Everything's the same. It's just rearranged. It's an isomer, as you just said, Rich. So she doesn't consider it modified in that same way.
Okay, that were semantics. I would say it appears in RNA in nature because of a modification. Okay.
Right.
But I'm pretty sure that in the case of making these RNAs, they have a precursor in the synthesis that's pseudouridine triphosphate. Okay, that it's not a post-transcriptional modification. And there's no reason to suspect that it wouldn't be incorporated just as well as uridine.
So they worked on a number of vaccines. They developed the lipid nanoparticle formulation. They showed it was also a good adjuvant as well as protecting the RNA, but they didn't do trials. Zika, as we said just before, it was over before there were enough people around to do a trial. So they were unable to really get these in people. They worked on influenza virus. And then, of course, brings us to January 2020. The new coronavirus emerges, sequences available in January. And so both Moderna and BioNTech immediately began to make mRNA vaccines. And they say here, mRNA vaccine candidates moved from proof of concept to EUA in just a year. How is it possible? Well, they already had this technology in-house, of course. Moderna, though, was a small company, so they got money from the U.S. They wouldn't have been able to do it on their own. And BioNTech, also a small company, made this deal with Pfizer, who big company, tons of money. They funded development, the clinical trials, manufacturing. So two different approaches for small companies to move this forward, one governmental and one private sector. Quite interesting. And as you know, the rest is history. And they say here, although there are many heroes in this story, Carrico and Weissman led the way to a rapid generation of vaccines by creating a safe and effective platform. This technology has revolutionized vaccinology. So, folks, we have a revolution that's happened just in the past year. I think we've appreciated it over time.
I certainly have. I did one of the ASV town halls, which are still ongoing, by the way, last week, and I got all wound up towards the end of it because it's amazing to just live through this and experience it. It's too bad that there are so many people out there who cannot appreciate or who won't appreciate the positive aspects of this, because it's remarkable what humans have done. It's incredible.
Yeah, I think the one thing that really struck me when I was reading this this paper was how excited I was to kind of have all of this written out and have all of these references kind of chronologically organized in one place so that I can then go through and sort of reread them all myself. And I found that to be really useful. But it was also really interesting to kind of think about this process of science and sort of where I was and what I was doing in those same years, and realize just how recent this was.
Yeah.
And that, in fact, I was working on DNA vaccines through most of this time, and I had no idea this was happening. And it was just sort of really interesting to have the story and the history written out so that I could think about how recent it really was and how revolutionary it really was.
Is it safe to say, Brianne, that DNA vaccines have not fared as well as mRNA vaccines?
At this point, absolutely. We have not seen any situation where a DNA vaccine has really made it as far. And so
why is that? Do we have any clue? Is it because mRNA is right there. It's translated. DNA need an extra step. Is that part of the issue?
I would think so.
I guess that's what it is. I still have trouble wrapping my mind around that, honestly, because if you had asked me during some of these very years of whether I thought an RNA vaccine or a DNA vaccine made more sense, I would have said a DNA vaccine because the RNA would get degraded too quickly. RNA is just not stable enough. That's why it needs to be a DNA vaccine. And so I was really blown away as all of this was happening to watch this awesome progress in RNA vaccines.
It's amazing.
So as I was looking at this today, of course, this celebrates the Lasker Award for these two individuals. And I'm thinking, down the road, Nobel Prize. Okay. And, it's Nobel Prize season, by the way. We're about to move into October when all of that stuff drops. But then I'm thinking to myself, Nobel Prizes usually, they have to endure some sort of test of time. They're usually, it's very seldom that they're awarded sort of short term. And then I ran across a Facebook post by my friend Lou Hammersholt, an HIV virologist, okay, who quotes from Alfred Nobel's will, which establishes the prizes. Get this, quote, all of my remaining realizable assets are to be distributed as follows. The capital converted to safe securities by my executors is to constitute a fund, the interest of which is to be distributed annually as prizes to those who, during the preceding year, have conferred the greatest benefit to humankind.
Yep.
So the original intent of the award was to make those awards for people who had done something very recent. Interesting.
Yeah, I think that obviously that's changed. Yeah, but Erling Norby talked about that too, how the original intent was to get actually relatively young people to help them, because it was hard to get support for science in the beginning, right? When the Nobels first came out, with no NIH and so forth. So is the Lasker a kind of indicator of Nobel? Is that?
Yeah. It's usually kind of thought of as the pre-Nobel.
There's another Canadian award. I think it's Canadian. That's a sort of a stepping stone to the Nobel. I forget what it is.
There's another one at Columbia, the Horowitz Prize. That's also kind of a Nobel indicator. I remember one year Stanley Prusiner got the Horowitz Prize, and he came to Columbia to deliver the address. And that day he got the Nobel Prize. He was up on stage and he said, well, they say this is a precursor, and they got it right this time. A precursor, I don't know, whatever. many of the people who get these prizes go on to get Nobels. All right.
Or already have them.
Have them.
If you look at the list for this year, since the other Laskers this year were for optogenetics and to David Baltimore.
Yeah, and as they say at the end, this is going to impact human health for forever. Given the broad impact, I mean, this is just not just a polio vaccine or a flu vaccine. This is going to impact way more than the pandemic. I would say it's Nobel Prizeable, right?
Yeah, I agree.
We'll see. But there has only been one Nobel Prize for a vaccine. That is Max Tyler for yellow fever vaccine. And the polio vaccine never got a Nobel. They gave it to John Enders, Weller, and Robbins for growing virus in, non-neural cells, which enabled you to make inactivated virus vaccines, but nobody else has gotten it. So I bet they don't give this for the vaccine, but they give it for the technology of making mRNA vaccines in general.
Right.
All right. Now we have a paper in the journal ISME journal. What does it stand for? Does anyone know?
Yes. International Society for Microbial Ecology. Although, at least I think, although the first, yes, I did confirm that. The first hit I got was for International Society for Music Educators or Education. So
this is part of the Nature stable of journals, I think.
Yeah.
Right? It's Nature. Okay. It is incomplete tricarboxylic acid cycle and proton gradient in Pandoravirus massiliensis. Is it still a virus? And these are a group of authors from Marseille, different institutions in Marseille, also one in Brazil, one in the UK. And the first two authors are Sarah Aherfi and Jamal Brahim Belhouari. And the last author is Bernard Lascola. And this is a group containing many other authors who have been working on giant viruses for many years, including Philippe Colson, Bernard Lascola, and the infamous Didier Raoult. Seems to be on all of these papers. Didier was involved in hydroxychloroquine, folks. You may remember that. So this is giant viruses we haven't talked about in a while. We've kind of been distracted by COVID pandemic.
Goings on. Yeah.
We used to talk about them all the time. In fact, Rich and I have been to the giant virus meeting, which happens every other year in Germany. We're there in 2019, right? Is that correct?
Yeah, that was pre-pandemic. That's the dark ages. I don't know.
Remember, yeah.
I don't know what happened before about Christmas 2019, but I think you're right. It was like actually quite soon before. It was like in November or something like that. 2019.
And there was one scheduled for 2021. It was canceled. So hopefully next year. They're fabulous meetings. All people interested in giant viruses. First discovered the Mimi virus, right? The first giant virus. Although there are pretty big viruses before that, which are now included as giants, like the pox viruses, right? And the virus Jim Van Etten works on,
Chlorella virus.
Chlorella, they're pretty big, but they're dwarf.
They're pretty big. They're about, so we when I was growing up, I used to refer to pox viruses as the largest DNA containing viruses around, which at the time they were, or known. Okay. And that's 200 kilobases. That's 200,000 base pairs in length. And, a gene costs you about a kilobase. Okay. So that's about 200 genes. All right. I think Chlorella virus is about 300. All right. And some of these giant viruses are what? Over 1 million.
Yes.
Okay. So when they hit the scene, everybody was blown away. They thought we had topped out at pox and Chlorella. Okay. But these guys are huge.
Yeah. They say here, virion size larger than 200 nanometer. What's the pox? Is it bigger than that?
350 on its long dimension. So what's this?
200. Well, they say that's the definition of a giant. Bigger than 200, genome greater than 250,000 bases. And so Pandoravirus salinus, which we talked about a long time ago, they say, broke all genomic records with a genome size of 2.5 million base pairs, 1 micron diameter viral particles.
So,
and then, yeah.
I was just going to ask, with that definition, so pox would fit that definition, right?
Yeah.
Which?
The 200 nanometers genome size greater than 250 kb.
Yes.
Yeah. So basically,
well, it's foulpox maybe over 250. Okay, vaccinia is 200. Okay, so some of the pox viruses break the 250 barrier, but only barely.
Okay, because I know that I've also seen sort of this definition of large DNA viruses that replicate in the cytoplasm and sort of make their own factories here. And so
NCLDVs.
Exactly, right? NCLDVs. And so that's sort of in the same range as this definition, I think.
What is that? The nucleocytoplasmic large DNA containing virus.
Yes, exactly.
Rich?
Yeah.
Because some of them are nuclear, but some of them shuttle. They seem to have a little. So many are cytoplasmic, like the poxies, right?
Right.
But then some of them seem to have a nuclear bootstrap phase that they need with that, right? So that's where the end. So there's a whole bunch of viruses. I'm not sure everyone would agree on this definition because the Marseille group likes to make their own definitions, and they get other people pissed off, like Eugene Koonin gets pissed off at them. And at these meetings, they yell at each other. And Koonin will get up and say, "No, it doesn't show that at all." And then the French people are up on, "Yeah, it does, Eugene. It really does."
Wait, it makes you angry, Rich?
Yes. Well, okay. Look. Nature is a continuum. Okay. And we're obsessed with drawing boundaries inside that continuum. Okay. Now, lots of times we do this for legitimate reasons. Okay. So that we can describe things and ascribe to them properties with some degree of generalization. And that helps us. Some people draw boundaries for the sake of staking a claim. Okay. For egotistical reasons. Okay. For, saying, I discovered this, or I named this, or this is special and I discovered it. Therefore I am special. Okay. And the work of some of the people associated with the giant viruses is replete with that stuff. And it drives me up a wall. Okay. And this paper, I have more highlighting than usual in this paper. I could go through it. About every other paragraph has some claim as to this being, a fantastic revolution in, our concept of what a virus is. And I'm saying no. Okay. So it drives me nuts. And I would go back to one person that we already mentioned, which is, I can pronounce his name, Didier Rouault, our hydroxychloroquine friend, okay, where you see this kind of hubris play out in a fashion that's really damaging, okay, because he made lots of what turned out to be false claims about hydroxychloroquine, and I think really contributed to a lot of harm that was done in the process of the pandemic. And my opinion is that this was done not for scientific reasons, but for egotistical reasons. And that drives me nuts. Science needs to be done for science, not for ego. So there's my wrap. I can go through my highlights later if you want.
I mean, the title, right? It's
is it still a virus? Baloney.
Of course, it's still a virus, right? It still needs cells, right? It doesn't grow on its own.
And I mean, the last sentence of the abstract is the one that really bugs me because it doesn't make sense in a syntax way, and it also is getting at that exaggerated claim type of thing. So that last sentence is, the presence of a proton gradient in P. macilliensis makes this virus a form of life for which it is legitimate to ask the question, what is a virus?
Or the last paragraph of the whole paper. This is preempting this. We can get back into the real science because there is science here that's interesting. Okay. In conclusion, P. macillensis undermines the last known historical viral hallmark, the lack of the Lippmann system. Thus, our findings presented herein raise critical questions concerning whether Pandora viruses can still be accurately classified biologically as viruses and renew deep-rooted arguments regarding the living nature of viruses in general. BS, 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 BS.
I am so glad I'm not the only one who.
This has been driving me nuts for years. Okay, and I'm glad that I can now express my frustration to in excess of 10,000 individuals.
It is quite clear that many of the, what they do in the introduction is go through all of these characteristics of viruses and knock them down one by one, right? And that's fine, but it still remains a virus because when it's not, then it's a cell.
Well, right? Also, I feel like there's one other pretty big characteristic that I usually teach that they just ignore because it wouldn't be able to be knocked down here.
What, they need to get into a cell?
That they can't do translation.
Yeah.
Right. But, they make the point that now some viral genomes, 2-pan virus, have almost a complete set of protein synthesis apparatus. They encode all the proteins but the ribosome.
Yeah, so they don't have a ribosome.
They still need a cell. Yeah.
Yeah.
So that's great that they have all these other proteins. I think it's really interesting. And, metabolism genes and others. But in the end, it still needs a cell. And if it didn't, there wouldn't be a virus, but it'd be a cell. So I don't get this at all.
To me, they cite all these examples. There's viruses that have some of these translation factors. There's viruses now that have some of these TCA cycle genes. There's viruses that have this or that. But it doesn't mean there's not one out there. But we have yet to see a virus that has all of these things and generates ATP and translates proteins.
Yeah, I feel like this comes up in microbiology a lot about why obligate intracellular parasite bacteria like chlamydia and rickettsia still get to be called bacteria and aren't called viruses. And the very obvious line is the ribosomes. And here there still is no ribosome, so it seems to remain clear.
At these giant virus meetings, most people are pretty sanguine about this. They say, these genes are there to modify host processes to favor virus reproduction.
Yeah, so, and that's cool.
Exactly.
That's
cool. Great.
Makes a lot of sense.
Super cool.
And probably what we're going to talk about now, the energy production, is the same thing.
The thing is, you don't have to pump this up. It's cool all by itself. Okay.
No. 1 year, not the last meeting in the giant virus, but the previous one, I'd invited one of these authors and another group in Marseille. They don't get along. There are two groups in Marseille that work on giant viruses. It's very funny. Chantal Abergel and what's the guy's name, Rich?
Clavier. Claverie?
Claverie. Jean-Michel Claverie. And this group here of La Scola and et cetera, they don't get along. So I had put together one with Claverie and Chantal and one of these guys, and they came up to me at the meetings. I can't be on the same panel as them. I'm sorry. I just can't do it. He didn't say why. Just, in French, I just can't do it. So, okay, it's fine. So I grabbed Jim Van Etten, and he was great to have on it. But to the extent that you so disagree with someone that you can't be on a panel with them, it's too bad, right? That's kind of counterproductive. All right. So they go through. Here's another thing that really bugs me. They say, among scientific discoveries causing giant viruses to challenge viral definition, which is BS, as Rich said, were the findings of associated virophages, which depicted for the first time a virus being infected by another virus. And that is just BS. It is a helper virus. It depends on a helper virus. These virophages need to be in the same cell as the helper virus. They don't infect the virus.
Right.
Right? Wouldn't get anywhere if they infected the virus. I don't know why they say that. It's kind of misleading.
Because it makes them cool. Because they discovered this.
anyway, so we have actually talked on TWIV about some giant viruses that have metabolic genes, like fermentation, even lipid biosynthesis, nitrogen metabolism. And they say, okay, what's left? The ability to produce energy. So that's what this is about. And what's interesting about these giant viruses is they say are, in this particular Pandora virus, Mesiliensis, over 80% of the predicted open reading frames are what they call orphans, open reading frame ants, or fans, which means it codes a protein that we've never seen before. It doesn't look like anything. It doesn't have a motif that we can say, oh, it's an enzyme. It's got an active site. None of that stuff. So there's obviously a lot to discover, right? So they look for proton gradients because, as they say, energy generation in the living world is associated. Life is a proton gradient, really, in the end, right? Somebody said that a long time ago. It wasn't me. So they have dyes that they use here and antibodies. There's a dye. One of them is called MitoTracker, which is a red fluorescent dye that stains mitochondria, and its accumulation is dependent on membrane potential. Okay. It's considered to be the name tells you, right? MitoTracker, tracking mitochondria. And there's another one called TMRM, is another dye that's sequestered by active mitochondria. So both of them are staining active mitochondria. So they do an experiment where they look at not only virus-infected cells, but virus particles. And they say 20% of the total number of virus particles were labeled by both antibodies to the particle to show that they're virus particles and MitoTracker. And they're also labeled by this TMRM staining. Their positive control is Staph aureus, which of course stains positive. There's no mitochondria. What are in those?
They themselves have a membrane potential.
They themselves without having any organelles, right?
They
went into eukaryotes to become mitochondria.
Yeah, so across the plasma membrane, they have a mitochondrial. Right. They have a membrane potential.
So what actually sets off MitoTracker? Is it a high concentration of protons or something like that makes it go red?
Membrane potential. Yeah, proton gradient across membranes, I believe.
Yeah, I'm double checking that now because I think that I've seen it before as a thing that tells if a mitochondrion has lost membrane potential.
I think it doesn't stain if it has.
Right, exactly. So that's kind of what I'm trying to figure out.
But I don't know how that works.
I know the interpretation is that the particles that stain have a membrane potential, specifically a proton gradient. Is that right? But I don't know the mechanism.
It just says it stains cells with a mitochondrial membrane potential. It doesn't say anything about protons.
Membrane potential is not necessarily protons, right?
Yeah, sure.
That's any sort of charge differential between inside and out.
So basically, the compound goes into the cell. The cell, if there is active respiration, then the compound gets oxidized.
That's it. It's oxidized.
All
right.
Okay.
So, Rich, their negative control is pox virus, right?
It's cowpox, yeah.
Cowpox, where it doesn't stain, not making any energy. These are the virus particles, right? And they also looked in infected cells, right? They saw staining with MitoTracker and also antibodies to the virus. So within the cell, they have membrane potential as well. So what do we think of that? Are we convinced by this? It was a pretty simple experiment.
Yeah. I mean, the pictures are great.
Good.
Okay.
Yeah, pictures look
it's
good.
Not 100%. It's like 20% of these particles, but they're lit up. So I'm thinking if oxidation, that's consistent with, in my sort of pretty rudimentary understanding of this kind of stuff. This is the area in sort of chemistry, biochemistry, where I get really on thin ice. But if it's oxidized, I think that's consistent with the notion that there is in fact, you're sensing a proton concentration difference.
Yeah.
Yes.
Okay?
Yes.
Okay. So that is consistent with a proton gradient. So yeah, I mean, I thought the pictures are beautiful. Okay.
Yeah,
they say it's about 20% of the particles. I'm not sure what that means.
Yeah.
Okay.
Yeah.
It could, but certainly in some significant fraction of particles, they're lighting up with MitoTracker.
Yeah. And I like the fact that they made antibodies against the particles so that they can distinguish between sort of the host mitochondria and the particles themselves. I thought that was a useful part of this experiment.
And I think cowpox is a reasonable control. I would like to see other giant viruses.
Yeah, it'll be cool.
What they do. Okay.
They also show infected amoeba. This virus can infect amoeba. So they stain with MitoTracker and this antibody to the Pandora virus. So you can see a few of the particles have red MitoTracker in them. But you don't see, it's interesting. I guess, so where would making energy be important? I guess in the cell, right?
Mm-hmm.
Yeah.
Maybe early on when the particle gets in, it could make some energy and that could be useful, or maybe.
Yeah, it doesn't logically make a lot of sense to me that there would be this gradient in the particles. Okay. I mean, I can see the virus tinkering with the cellular mitochondria or something like that. Mm-hmm. How or why you wind up with a membrane potential in the particle itself. I mean, these are probably finished particles, I would assume.
Yeah.
And so that doesn't necessarily make a lot of sense to me.
Yeah, I think that there are a couple things that I don't know enough about to fully be confident. For example, I do not know a whole lot about amoeba immune responses. But what I do know in terms of thinking about sort of mammalian immune responses is that there's this growing literature of immunometabolism and the idea that different compounds that are made in these metabolic processes can modify immune responses. And so my question is the membrane potential the cool thing here? Or does the virus want to make some molecule to mess with the amoeba's response? And just in the process of making that molecule, they happen to make a membrane potential.
Yeah,
and sure. We're kind of at the edge of my knowledge at that point. So there's not much more I can say about that.
Yeah, that makes sense. Yeah. It's an accident. But of course, maybe this is ancient and that became anyway. So like all good experiments, they have an inhibitor. It's called CCCP. Doesn't have anything to do with Soviet Union, I think. Carbonyl cyanide M-chlorophenyl hydrazone, chemical inhibitor of oxidative phosphorylation. And they use different concentrations, and at each concentration, they incubate the particles, purified particles, with this inhibitor, and then they add the TMRM, kind of the MitoTracker, the other MitoTracker, and the signal goes down. So that's good. That's consistent with oxidative phosphorylation happening there.
Well, it's consistent with there being an electrochemical gradient.
Yeah.
Okay. And I spent a lot of time trying to figure out, once again, the mechanism of action of CCCP, and came up more or less empty-handed, other than it destroys chemical gradients. Whether that has to do with messing with membrane permeability or a particular OXPHOS enzyme itself, I could not understand. Okay, but I think that and maybe we're going to get mail here, and that's great. Okay,
that's fine.
But my fundamental understanding is that it destroys an electrochemical gradient, which doesn't necessarily have to be even protons. Okay. It can be any ion. Okay. And so I don't know that the CCCP experiment specifically fingers oxphos. Okay. Or if it's more generally that it implicates an electrochemical gradient.
Well, I just looked it up and it said it's an inhibitor of oxidative phosphorylation.
Right.
So,
yes. And that's where I started to try to drill down deeper and say, okay, why? Because if something, for example, messes up membrane permeability, that'll inhibit oxphos. Okay. Sure. Simply by destroying the proton gradient.
Yeah.
Okay.
I mean, they could have used, for example, cyanide, which targets what? Cytochrome.
Right. Exactly.
Right. And maybe that's too specific. And this system doesn't.
Exactly. Let's do some more drugs. Let's do some more experiments. Exactly. And find out exactly what this is.
I mean, the one thing in their favor on this specifically messing up membrane potential is that they use this drug TMRM that is a membrane potential measurement.
Mm-hmm.
And that is what goes away. But everything that Rich said is completely correct here.
I think the experiments do show that there is an electrochemical gradient across these virus particles. Yes. Okay. But what the nature of that electrochemical gradient is, or why it's there, or what virus proteins, if any, are involved in generating an electrochemical gradient, I don't know from this paper.
Then they take these virus particles that they've treated with CCCP and they measure their infectivity, and there's no change. You get the same titer, TCI 10 to the 7.2, TCID 50 per mil. So getting rid of this gradient doesn't affect infectivity, which I'm surprised in a way because I would think that's when it would help, but maybe this is not the right host. I don't know. Right?
Well,
it's not clear to me that amoeba are the right host for these big, they grow in them, but it's not clear it's a natural host, right?
Yeah. And I think that's the other question I have. And this, again, is just more of at least partially my not understanding of every last detail here is they got rid of the potential before they infected the amoeba, but they don't show whether they can sort of remake a membrane potential when they're in the amoeba.
Mm-hmm. They did say, Bria, they say we could only observe a lower number of viral particles on amoeba cells infected with virions pre-incubated with CCCP at time 0 and time 3 post-infection.
Right,
so
but
early.
But they don't say that the virions maintain a loss of membrane potential. So could they rebuild it when they were not in the presence of drug?
They interpret that to mean that it's involved in an early phase.
Right,
infection.
exactly.
Right. And this is, they say, chlorella viruses, they have potassium channels, apparently. And if you inhibit those, it prevents DNA release. Also other viruses, bacteriophages, and so forth. So, all right. This is an interesting paper because they have results and then they shoot off into discussion, which to me belongs in the discussion section.
Yeah, this is kind of a results discussion mashup.
Yeah.
And then a conclusions section. Yeah.
Yeah, that's right. It is a mashup, which is fine because now they cite all these different examples of viruses where these different activities have important roles and so forth. I mean, the bottom line is, as they say, viral communities can adapt the diversity of their auxiliary metabolic genes to exploit and master their microbial host. That's what they are, auxiliary metabolic genes, right? They're tweaking the host to make reproduction a little better. Then another set of experiments, they add acetyl-CoA to the virus particles. Guys, people here, of course, remember acetyl-CoA. It's the thing that comes into the TCA cycle, right? The Krebs cycle. All that circle of all the things, all the compounds being converted to one another that you had to memorize in biology in school, and little energy is made at different times. So acetyl-CoA is the input. So they add either, what do they do? They add the signal with this TMRM goes up when you add low concentrations of acetyl-CoA, And it goes down when you add high concentrations. And it's the same result with Staph aureus, with low and high concentrations of acetyl-CoA. So I guess the idea is that you put a low amount in, it's used, and you get more membrane potential. But if you put too much in, it's inhibitory.
Basically, the way I see it is that experiment links the TCA cycle to the electrochemical gradient that we've been talking about before.
That's right.
Okay. Because I said previously, the experiment showing that there's an electrochemical gradient all by itself doesn't necessarily tell you where it's coming from. But the acetyl-CoA experiment suggests that electrochemical gradient is related to the TCA cycle. Okay. I'd like to see more experiments, more inhibitors, et cetera, but
they say this highlights the possible existence of a TCA cycle in Pandora virus.
Right.
All right, and we'll see a little bit more evidence for that as well. So then they do bioinformatics. Let's see if we can find some genes encoding the genes that we know are involved in the TCA cycle. There are a lot of them that go around that circle. I remember my high school biology teacher, she drew it. And she never mentioned ATP or any other energy. She just drew puffs. She said, you get a little puff here. And I was confused for years. I wish she had just said, you make a chemical that has energy in its bonds. That would have made much more sense to me. What the hell are the puffs? Because then I go to college and I'm lost for a while. I said, where are the puffs? So they find a low amino acid sequence similarity with some enzymes involved in the TCA cycle, basically. Let's see, they found 8 or so, right? 8 different p-mesylensis orphs, citrate synthase, aconitase, isocitrate isopropyl malate dehydrogenase, and then they give you the viral gene names, alpha ketoglutarate decarboxylase. You may have had to memorize all of those at one point in your life.
I managed to avoid that.
Then you quickly forgot them. Do you remember years ago, metabolism in viruses suddenly became hot? I think Steve McKnight kind of was at the spearhead of this. Okay. He found that viruses are tweaking metabolism, as did Tom Schenk and others. But I once was on a flight and he was next to me. I didn't know it was him. And I was reading a Cell paper and he looked at it and he said, are you a scientist? And so then we introduced each other. And he said in our conversation, he said, I thought I had forgotten the TCA cycle forever and I never had to deal with it again. And then we find that viruses are playing with it. I'll never forget that.
But that's a good point because there's precedent for viruses tweaking energy metabolism.
Acetyl-CoA synthetase is just upstream of the first step. They also have an ortholog. So these are proteins encoded in the p-mesylensis genome that have some similarity with these genes. And then they go back into their discussion and talk about, they don't all have to be the same because they said maybe these should be all present in all the giant viruses, but no, they could be different. They cite some variant TCA cycles in different organisms that we know about. Okay, so now we have these orthologs, and they talk about the TCA cycle being a hub of metabolism. If you don't remember, it produces energy by oxidation and eventually harvesting electrons, and those electrons make your proton gradient across the inner mitochondrial membrane, and that gradient makes ATP synthase. And it also makes a lot of intermediates for other processes, this TCA cycle. It's quite versatile, like precursors of bases, fatty acids, and so forth. And this is really important. I've learned this from TWIM. You don't have to use oxygen as a terminal electron acceptor. You can use a lot of other chemicals. You don't need oxygen, and you can make plenty of energy. Okay, now they look for ATP in the particles and they didn't find it. This is data not shown because obviously if there were a TCA cycle, you might predict that you'd make ATP. They didn't find it. And they say, well, maybe there are alternative metabolic pathways that we don't know about. And then they talk about the origin of the TCA cycle, which I don't really want to talk about. It's kind of meandering. And okay, so we have enzymes, we have proteins with some homology. Then they look at the transcriptome, the RNAs that are made during virus infection. This has actually been published before. And they ask, do any of these 8 supposed TCA genes, are they transcribed? And 6 of 8 of them are transcribed at different time points. And they find, between 8 and 16 hours is a prime time for some of these open reading frames. All of these TCA ORFs that they call them were transcribed at the same time points, especially at the end of the developmental cycle, which is weird, right? At the end. So does that say they're probably not participating in reproduction? There may be, I don't know, put into virus particles for the next cell. I don't know. It's odd.
Yeah, or somehow involved in, I guess, somehow release, maybe.
Yeah, maybe.
Yeah, but this is one of,
look at that. Yeah.
Yeah, this is one of those things that made me wonder. Well, then does it matter if you inhibit it at the beginning versus inhibiting it at this later time point?
2 of the 8 orthologs are found in viral particles: the putative alpha-ketoglutarate decarboxylase and the putative acetyl coenzyme A synthetase. Just 2, though. I mean, so you probably don't have a complete TCA cycle in the particle, right? If you don't have all of them. I don't know.
Or is it just that they couldn't find them? I mean,
maybe they couldn't find them.
It's a negative result.
Yeah. They produced some of these genes in E. coli. They made the protein, and they looked to see if they could measure the activity. Boy, I wouldn't want to be doing that.
Oh, they just bought a bunch of kits from what I could tell. They bought
kits?
Reading the materials and methods. They bought kits for this, that, and the other enzyme. Yeah.
They couldn't get any enzymatic activity for most of them, maybe because they don't have the right conditions in the kits, right?
Maybe.
Except for one isocitrate dehydrogenase for 2 open reading frames. And they say they're pretty low activity. But they say it's probably not just chance that this isocitrate dehydrogenase has activity, right? It's probably real. So that's good that it has some activity. So that's it. They have proton gradient in particles. They have, it's inhibited by inhibitors of such, stains with mitotracker. They have 6 of 8 TCA cycle gene orthologs. Some of them seem to be transcribed. Some of them seem to be in particles. I think it's interesting that all of this, right? And need a lot of work, obviously, still, but I think it's quite interesting.
So there's a couple of things here that strike me. First of all, it doesn't have the complete TCA cycle. There's a couple of enzymes missing. Yeah. The enzymes that are there, the homologies are apparently not strong. Okay. They tried to express them all in E. coli to see if they could get activity. Only one of them showed activity, the others didn't. Okay. Yeah. To me, this says that at some time in the evolutionary history of this virus, well, certainly it probably acquired these genes from cells. I would think, though you could debate that. But I think that's a logical, some sort of horizontal transfer that acquired these genes. They may have, during some point in the evolutionary history of this virus, been handy in tinkering with intracellular metabolism. It may still be true in some circumstance that it does, but it's not shown here. Okay. So I think all that is, quite interesting. As we've already said, I think the conclusions go way beyond that into spaces that I don't think are justified. But that doesn't mean that the data themselves is really interesting. And I'm reminded of one thing This just came to mind. I don't know, 30 years ago. What would be early 1980s? That's 40 years ago. When Niles and I sequenced a chunk of vaccinia virus DNA. Back when you would blast something, you'd get no hits for anything because there wasn't anything in the database. It was all dark matter.
Yeah.
Okay, and we had, I don't know, what, 12, 13 genes. And I remember blasting all those when we finally got the sequence. And the only one that gave any kind of a hit that was just a blistering hit was for carbonic anhydrase, except that it had a membrane anchor on it. It was and carbonic anhydrase is an enzyme that interconverts CO2 and carbonic acid that's important in CO2 exchange in the blood. It's contained in red blood cells and a few other things. So carbonic anhydrase has absolutely no business whatsoever being in this virus, in particular with a membrane anchor. Okay. And in subsequent years, it was shown that it didn't actually have carbonic anhydrase activity despite the really strong homology. And that enzyme, it turns out, or that protein, it turns out, plays a role in attachment of the virus to the cells. Okay? So it has nothing to do with carbon dioxide metabolism. So who knows what these things do, though the fact that you have a whole raft of them is provocative, I must say. And
it could be that the cell is providing the missing links, or as they suggest, maybe it's a totally weird Krebs cycle, TCA cycle that we don't know about. Yeah.
Of course, what's really missing, and it's not their fault, is knockouts. You need to be able to go into the virus and knock these genes out and infect cells with them and ask what happens. Okay. Do you still see all these phenomena with mitotracker concentrating? Does it affect the virus growth and that kind of stuff? But that's for down the road. And I don't think that's even technically feasible at this point.
They kind of make an argument toward your dark matter thing, Rich, where they say you have to note that there's a limit of the standard comparative genomics tools for identifying novel or small or very phylogenetically divergent sequences.
Yeah.
And so maybe they're just not looking the right way, but they do say that if they can look at the crystal structures of these putative, produce these proteins and look at their crystal structures, that might be informative.
Right. Yep. Yeah. A lot to do.
Mm-hmm.
Yep. Cool viruses.
Very cool.
And, it just reemphasizes that viruses have all kinds of tricks. All right. And they're nothing is out of bounds, except maybe translation, Brian.
Yeah, I think this is just so cool to see the different things that a virus can do and to think about ways that, this might be really advantageous for this virus. But I still squarely seems to be a virus as far as I can see.
Viruses need the cytoskeleton and they need to assemble somewhere, right? So they're not going to just be dividing on their own. So they're viruses for sure.
Yeah.
By the way, if listeners want to go back and listen to about, the first 100, 150 episodes, there was a raging debate in TWIV as to whether or not viruses were alive.
We did.
That even at one point resulted in us taking a survey. Okay.
Yes, that's right. Yeah. Of listeners, right?
Yeah, of listeners.
And it was split, I think.
Yep.
Between yes and no. Yeah. Yep.
Two other comments. One that Vincent reminded us that Trudy Ray wrote about this in the virology blog, December 3rd, 2020. And the title is Pandora Viruses Are Not Alive. So she came down strongly along those lines.
Yeah, it was a nice article.
Yeah. And then I just want to have a little bit of a mini rant about supplemental figures and material, kind of in general, but particularly in this paper. First of all, there were 6 or 7 figures and a couple other tables or things, and you have to click on each one individually, and you shouldn't have to do that. You should be able to click on a single link and open all the supplemental material, in my opinion. And then most of them, once you opened them, you got some link that did not have the title or the number of the figure in it. Occasionally, in this case, the authors had put, supplemental figure 4 in the body of the figure. So once you had it open, you knew what you were looking at. But authors can do us all a big service by doing that. But journals can do us a big service by allowing us to download all the supplemental material at once and labeling all of it. It's like the, poor stepchild or whatever of the paper. And it shouldn't be if we're going to look at some of this stuff.
You also, Kathy, you put into the show notes some tomograms of Pandora virus. They came from elsewhere, right?
I didn't put those in, so maybe Vincent.
Who put those in?
Yeah, I found them. It's on the Marseille website. They have tomograms. Yeah, that's true. Very nice pictures, right?
Mm-hmm. So tomograms are these images where you're moving through the planes of the virus, and so it's kind of like taking sequential slices through it. They're pretty cool.
Yeah, I mean, they're very cell looking, right?
Yeah.
Small cell looking. There's no nucleus, but yeah, but why not? That's fine. Yeah. You're pretty.
What else is out there? You
just have to look, right?
Yep. Or get lucky. I mean, I forget the history of how this originally happened.
So the Mimi virus, was from a.
It's the one that came from an amoeba in a water tower.
So somehow it was associated with somebody who had an eye infection in a water tower. I don't remember, but they could see it under a microscope. So they figured it was a bacterium and they put it in the freezer for 10 years. And then they went back and they realized, I don't know what they did. They sequenced it or something, the genome, and they found out it wasn't a cell. So they called it Mimi for microbe mimic.
And once you open that door, there's a whole bunch of stuff on the other side. I remember from the Giants virus meeting, the guy from somewhere in Massachusetts who just dug a bunch of dirt up out in some, wooded area in Massachusetts and did metagenomic sequencing and came up with all kinds of these things. They're everywhere.
Yeah.
You could say instead of opening the door, you could say opening Pandora's box.
Ooh.
That's right.
You could.
Although apparently it's not a box, it's more like a flask. So
Well, and if they do this bioinformatics to look and see which other organisms have genes that are closely related to these ORPs and they don't find anything.
Yeah.
I think that just means that we haven't looked enough.
Right.
And I'd love to know whose genes are they actually similar to.
Right. All right. So here's the Mimi history to get it right. It was discovered in 1992 within the amoeba, Acanthamoeba polyphaga, after which it is named. So the original name was Acanthamoeba polyphaga Mimi virus. It was during research into Legionellosis by researchers from Marseille and Leeds. The virus was observed in a gram stain and thought to be a gram-positive bacterium. As a consequence, it was named Bradford coccus after Bradford, England, where the amoeba had originated. In 2003, researchers at Marseille published a paper in Science identifying the microbe as a virus. It was given the name Mimi virus for mimicking microbe. So the French renamed it. And they could say, we discovered Mimi virus, right? It's the same team that discovered it later discovered a slightly larger virus called Mama virus. And then subsequently Mumu. And at the last Giant Virus meeting, our host sang a song based on Mimi, Mama, and Mumu to the tune of, what was that?
I had gone to bed.
Oh, you went to bed. All right. What's the song? Mama, ooh.
Bohemian Rhapsody.
Bohemian Rhapsody. Thank you. By who? Who was the
Queen?
Queen.
He made the most amazing take on these.
Actually, I may have been around for that.
And actually, you sent me the lyrics, which I have as a text. He played the piano and he sang it. What was his name? Matthias
Fisher. Brilliant.
Oh, it was just
and
great.
talented.
That echoed in my head And then years later, I saw the movie about Eddie.
Bohemian Rhapsody was the name of the movie.
And every time they played it, I kept thinking of Mimi Virus. He was really good. You think you've heard it, Rich?
What's the song? You mean at the meeting? Yeah, I forget. I have a tendency to check out early, but I was there for some of the singing.
We need to get him to perform it on a TWIV episode.
Yeah, good idea.
Oh, it was so good. It's really so good. Anyway.
Matthias, get together with your buddies, make a YouTube of that. We'll link it on.
That would be cool.
TWIV.
All right. Let's just do one round of email here. We haven't done an email in a couple of episodes. Rich, can you take that first one?
Jerry writes, first and foremost, thank you for all the wonderful service and education you're providing all of us who are just things other than infectious disease experts. Just things other than infectious disease experts. I, like so many, am addicted to the knowledge, the banter, and caring you display. You have come to feel like extended family to me. Good. That's what we're here for. I have always been bothered by the lack of experimental correlation between testing and the risk of transmission. I understand how contact tracing and other things can work as a proxy, but would love to see a direct experimental connection. I came across this article, and it gives a link that might be able to fill this gap. It claims to use sub-resolution microimaging and AI to be able to identify SARS-CoV-2 in ejecta from speaking or such. It's eerie having these tools pull such detailed information in the blurs, but that is actually one of the things AI is quite good at. First, I was wondering if there was some way to first have the system locate what it thinks is a virus, then contact transfer the droplets onto divided replication surface. Then you could see if there is competent virus at the places the AI has called out. They may need to do more different training to get the AI to match reliably. If that were to confirm the detection method, then you could add this as a testing experiment to see the direct correspondence from testing to competent virus in ejecta. For me, this would approach a gold standard for testing as a correlate of transmission. I don't see this as a basic testing method like they seem to suggest, but I do like their idea of looking for tools far outside the common set. Yours, Jerry. And I confess, I have not looked at this particular article.
So I think this is pretty cool. They describe this artificial intelligence coupled with microscopy method where they're somehow able to identify specific virus particles, not other viruses, and they can tell them from dust and all kinds of other things. And so they're talking about it being sort of a very fast point of use test for the virus. Preclinical trial was highly successful with a 96% success rate for SARS-CoV-2. So the listener who wrote in doesn't see it as a test thing, but wants to go one further and make sure that the things that it identifies are a virus are actually able to replicate. And that might be necessary, I suppose. But they say the advantages to this kind of test are that it's on the order of 1 minute. And the second is that there's no added chemicals or modifications to the samples. And so it's just able to detect it.
I seem to recall, I don't know if it was this technique or something related earlier that they were talking about deploying as, basically a breathalyzer test for SARS-CoV-2 that you could use at airports and et cetera, which, is brilliant. And I think Jerry's saying that, you could use this as an aid to study transmission, because we're always talking about getting samples and, RNA isn't good enough. Even antigen isn't good enough. The sensitivity of trying to do cell culture is just too cumbersome. et cetera, although it does tell you whether or not the virus is infectious. And he's saying maybe you could use this as the foundation for a study on transmission. And I totally agree.
Can you quantify you have to be able to quantify the particles, right?
Yeah, and you need a particle to PFU ratio.
Yeah, it's almost like you need to have some way to send out your ejecta, and then you're shooting it, and you can then look at the things while they're in the air and say if they're viruses, but have it shooting at like a 385 roll plate, where you can then catch what's been taken and put into cells.
And I think that's what Sherry's saying,
Right. You have to correlate which thing the AI called as a virus particle landed in which well.
And then see, did that, could and then try to grow things out of each well.
Yeah. But in a way, it also is like a fancy way of having SARS-CoV-2 sniffing dogs. Except that they're artificial intelligence to identify the virus particles. And so it's not that far-fetched in terms of being able to specifically identify something. But to go one step further and do the infectious assay might be a challenge.
I just think it's great that people do these things, right?
Mm-hmm.
Yeah.
They think out of the box, try different things. You never know what's going to hit. And that's how mRNA vaccines arrived, right? So it's great. Who knows where it's going to go? But very cool. I would never have seen this. Kathy, can you take the next one?
David writes, "Hi, Twiv. A lot of the controversy around the Wuhan Institute for Virology centers on their method for testing wild coronavirus spikes. There are two ways they do this: by putting the spike in a pseudovirus that's very safe, and by making a chimeric coronavirus, which is controversial. Do you think the benefits of testing a wild spike in a WIV1 backbone outweigh the risks? Why not just use pseudoviruses? We can make humanized mice sick with a WIV1 chimera, but are we really sure we're learning something about the virus that the spike came from? Or is this just so that we can check the in vivo box on a publication?" From the Wuhan Institute for Virology's released grants, the uproar is about the fact that they made pathogenic chimeras, but the benefit is that they revealed that there are coronavirus spikes in nature that we didn't know about and should be concerned about. The question is, could they have come to a similar conclusion with just pseudovirus assays? Thanks, David.
Well, from last Friday's paper, they just made pseudoviruses with all those spikes that they pulled out of bats in Laos. Remember, Rich, you were on that episode, and they concluded these can enter human cells. So if you want to know about entry, that's all you need. But of course, it's not everything. On the other hand,
but in the end, they took samples from those bats and showed that there was a virus that would infect vero cells. Okay, and that was the nail in the coffin for us, a virus that does it. So, taking it to the extreme experiment of making the virus is, to me, important. Though it's worth thinking. I mean, the letter raises a perfectly legitimate question: what added benefit is there to making the virus relative to the pseudoviruses? And I think there is probably some. My guess is the spike protein is displayed on the surface of the actual virus slightly different than in the pseudoviruses. It could affect the uptake. And if you really want to know whether, the spike plays, how much of a role it plays in the infectivity of the virus, you really have to do it in the context of the virus. And I would add that you can do all this stuff safely. Okay. Yes. We work on really, really dangerous stuff all the time. And that's what a BSL-4 lab is all about. Okay. It can all be done safely and responsibly. And to the best of our knowledge, that's what has been done. And so I think the popular press makes much too big a deal about the fact that experiments that sound dangerous are being done, because these are perfectly legitimate experiments that can be done safely.
Yeah. And I think that, the experiments with the pseudovirus really get you information about the specifics of sort of entry and the specifics of susceptibility. Yes, you can get the virus into this cell, but is this cell actually relevant for more details of replication? Needs to be done with the actual virus. There are lots of viruses that can get into a cell, but then can't complete their replication cycle within that cell. And so if you want to actually see if you can complete the full replication cycle, you need to have a full virus.
And if you can do this safely, I agree. You can do it safely.
If you decide that nobody can do any experiments that would be required to be done, that are dangerous enough to be required a BSL-4 facility, then science, this type of science is going to be set back on its heels for decades. Okay. You can do these experiments safely and they should be done.
Brianne, can you take the next one?
Sure. Bob writes, wow, after the thin gruel of journalistic science, in quotes, I always look forward to TWIV episodes, but episode 802 was so meaty. I just finished my third time listening. And 802 was the last episode we had with Shane Crotty. I may need a long nap to digest this. One thing I wonder about, given the uncertainty around the reported case to actual case ratios, have there been any broad serological surveys to figure out how many people have antibodies one way or another? I am just an armchair epidemiologist, but some of the case multipliers I have seen would put us near 80% exposed via virus and or vaccine and producing antibodies. That would put us just months from complete immunity. This does not mesh with everything that is happening, so I have to feel that we must be reporting cases more completely than we think. Thanks for all you do. I always feel ahead of the game after listening to you, having noticed that whatever skepticism or doubts you have about the latest headlines, you start getting expressed in the media about 2 weeks later. So thanks, Bob. And Bob, I would actually love to see those same data that you mentioned. Although the one piece that I often think about with this is oftentimes when we think about those multipliers, we are talking about those who are eligible for the vaccine. We're not counting all of the kids who are not eligible, which dramatically reduces the percent of our population who has at least been vaccinated.
I would add, I had a pick a couple of weeks ago of the Texas CARES project. I'm sure that there are many similar projects around. This happens to be the one that I'm familiar with, where they asked people to volunteer to take blood samples over time to test them for antibody to the spike protein and antibody to the N protein, which will allow them to distinguish people who've been infected from people who've been vaccinated. It can't pick out people who've experienced both. And there is there's a dashboard for this project. The enrollment is now closed. They managed to enroll 86,615 people. And of those, and this is Texas, and this is people who enrolled in the project. There's going to be selection there. Okay. But of those people, 24% were N positive. So 24% have been exposed to the virus itself. They have the data for who's been vaccinated but not infected, but it's not on the dashboard yet. I've corresponded with them and they said it will be eventually. Okay. I'd love to see that because I don't know what that number is. But yeah, I mean, if you got 24% who've been infected and we have, at least 50% or more of the population in Texas that's been vaccinated, there's going to be some overlap between those, but you're going to be, upwards around 70% or so, I would think, that are S antibody positive, maybe even more.
Yeah. I also think back to, do you remember the paper on excess mortality that we did where they said that's that's the most reliable indicator of deaths because everything else has got issues, including serological surveys. They can be biased and confounding and so forth. Like the one in Brazil, which involved blood donations, where it's not a random selection of the people who go give blood are not random. So It's hard. It's really hard these multipliers are not all right. And so that's probably why we're off also.
Yeah. If you're counting excess mortality, people aren't volunteering for that.
Right.
Okay. So there's no selection.
Right. It's of course different from just being infected, but it's just to point out how it's hard. Even with people dying, it's hard to calculate the effect of COVID unless you look at excess. And Lori writes, I am a nurse in Bozeman, Montana. I love your show. Listen to Daniel and the gang weekly. Almost. I really appreciate the education you are making available to all of us. We are giving more monoclonal infusions now more than ever in my town, and that is 10 a day. My question is, in 3 months, if these folks remain unvaccinated, will they be fresh meat for another infection? Will there be a post-monoclonal wave if they remain unvaccinated, as many have stated? Are they without antibodies? So let's take that one. I would say it depends when they got their monoclonal, right? If they got it really early, which is probably unlikely unless they're tested frequently and they're at risk, then they're probably not going to have much of their own antibody response. If they're given late, better. Correct?
Yeah, that's
exactly what I was thinking.
Given late relative to what do you mean?
When they were infected or when they showed up. Okay. Because I hadn't built that into the equation. All these people getting monoclonal antibody therapy have been infected. So how far was the infection progressed before you gave them the monoclonal antibody therapy? That will determine what sort of residual level of antibody or immune response or memory that they have. And I don't know. Do you know, Brianne?
Do I know what?
I think they're going to eventually be fresh meat regardless, because we think that if you get a natural infection, your immune response is not that strong or long-lived. So even if they did develop something at the end of their monoclonal treatment, that passive immunization is going to go away.
Yeah, so the passive immunization is definitely going to go away. And I agree that the one variable I'm not sure about is how much time did their immune system have to try to mount their own antibody response, their own B-cell response before these monoclonals came in and sort of shut everything else down. And the timing, I think, would matter quite a bit.
But it's not crazy to think that the monoclonals could compromise what would otherwise be, some normal natural immunity, right?
No, it is not. We know that pre-existing antibodies can actually inhibit naive B cells from responding. So those monoclonals would actually turn off the native B cell response. So it's just a question of how long that response had before it got turned off.
Okay, this is a vaccine question. Some nurses are saying that since there are more and more infections of the vaccinated, that there is no reason to get a vaccine. My thought is with masking, social measures, and vaccines, we slow the spread and decrease the infection rate. If we allow it to keep spreading so much, we are increasing the chance of mutation and slowing our get back to new normal way of life and work and play. And vaccines are safer than getting infected, possibly getting more seriously ill or having long COVID. Now, some other concerns nurses have is that the vaccine used fetal cells in the research. Is that true? That's not an issue for me, but these are some of the roadblocks. I listened to the show about young women and fertility concerns, so no question there. Keep doing what you're doing. I know it's hard. I'm burned out from this. And we aren't even overrun in my hospital. Very busy, but not totally overrun. We will get through it together. And again, I am thankful every day for the education from your shows. Okay, there's one more question from her. So I think you should read that article by Craig Spencer from last week that, the vaccines are still working really well. This idea that more and more infections of the vaccinated, it's still much less than unvaccinated, and you stay out of the hospital and you don't die. So there's that. The fetal cells, 293 cells were used way in the beginning when they made the mRNA. They put it into them and they said, the protein is made. We can go forward.
Right.
It wasn't used in the production, but it was used in the testing of the mRNA.
Yeah. And so just for people who are less familiar with 293 cells, they were used for both the mRNA vaccines, Pfizer and Moderna, exactly as Vincent said, just to see if protein production was made. And they both came from a fetus from the early 80s, I believe. I used to know the year. I don't remember the year anymore.
Long time ago.
Yes. And
decades.
The Johnson & Johnson is an adenovirus vaccine. It does use a different fetal cell line that's proprietary at Johnson & Johnson to as sort of a factory to put together the adenovirus. None of those cells are in the vaccine. They were used somewhere, either in a testing or as sort of the factory to make the virus.
So the fetal cells are no longer, they were in the early stages of research for the mRNA vaccines. They still get used in production of the virus vector vaccines, but as Brianne points out, none of that stuff is in the vaccines themselves, and no new fetuses are sacrificed to do any of this. It's all from decades ago, and the Pope says it's okay.
Right.
I'm not joking.
That's
the
true.
Pope specifically said it's okay.
It's one of the early things that popped into my head. The 293 cells are from 1973, and I think must be from Europe.
Yes,
Netherlands.
When I was up to date on it, I knew that both cell lines, the 293s and the others, were both from the Netherlands. They were both about a year apart.
Percy 6. There's some other ones you're thinking of.
Yes.
Okay, one more question. So we have a communal break room where we eat lunch. Montana passed a law this year that forbids an employer to ask employees their vaccination status. I asked some coworkers, and I know about 20% in my ER are not vaccinated. I mask when changing and eat outside, but winter is coming, and I will move inside. Any thoughts? Thank you, Twiv. I think if you're vaccinated, you wear your mask, you're good.
Except you have to take your mask off when you eat. That's her question.
Yeah, you might want to go off in the corner, keep your distance.
I would stay away, yeah.
Or
stay away.
Time shift, eat when there's nobody around if you can.
And if you want to eat with your friends, choose friends that are vaccinated. Well, if they don't want to tell you whether or not they're vaccinated, then don't eat with them.
You could blend your sandwich and drink it through a straw. That way you can keep your mask on.
Mostly, yeah.
Could you blend a sandwich? I guess it would be hard. Yeah. You'd have to put some liquid in. That would be not very interesting, right?
No.
Montana passed a law that forbids an employer from asking their vaccination status. So that overrides the HIPAA regulations. Because my understanding is that according to the health information privacy protocols, it's okay to ask somebody whether or not they've been vaccinated. They don't have to tell you, but you can ask, So this goes beyond that. This says you can't even ask. Crazy.
Can you require them to be vaccinated to work?
Actually, I think so.
You can, as long as you have some kind of religious or medical exemption, and how strict that is varies state to state.
So if an employer says you need to show your vaccine card to come to work, does that constitute asking them if they've been vaccinated?
Probably, yeah.
Does in Montana potentially.
So that gets around the whole need to be vaccinated, right?
Sure.
Mm-hmm.
Here in New York State, a federal judge has ruled that employers can require their employees to be vaccinated.
Right.
Oh, well. Okay. That is it for our questions. Let's do some picks. Brianne, what do you have for us today?
I have something from PhD Comics that I saw recently, and I had to laugh rather hard because this is a list of sentences that it says are said by no academic ever. And I agree on most of them. I turned in my grant early. I don't have any comments. This meeting was a great use of my time. Things like that. I looked at all of these and I said, yeah, they kind of hit it right on the head here.
Actually, I have said specifically number 4. I don't feel I really contributed. You can take my name off the author list.
Okay.
Yeah, I have done that too. Yeah.
I think I've said it's the weekend. I'll reply on Monday when I really didn't want to do something.
They're good.
You never loved the conference coffee, right?
No.
It's never any good. No. That's why everybody goes to another place to have coffee.
I love this. I'm all caught up and have nothing to do.
Yeah.
Is what I like. That faculty meeting was a great use of my time.
Exactly. I thought this really captured many of my feelings on
it's really
different
good.
things.
It's really good. Like it. Very, very good.
Thanks, Brianne.
Kathy, what do you have for us?
I picked something that was given to me by my friend Sarah, and it's an entree into Alex Chen's work. And this particular one is a Bach Cello Suite No. 1 visualization. And Sarah learned about this whole person's work through a data visualization course. But this one draws lines for that are the lengths are proportional to the frequency of the note, and then it plays through the Bach Cello Suite. And then there's several other really interesting visualizations. There's one that's the subway map of New York City. That one also happens to be music. What were the other ones? Oh, there's a Piano Phase. That's another music one. Harmonics, beach balls, crayons. And so this person is creative director at Google. So he's the one who's led to some of the Google Doodles and things. But these are just, this is kind of a black hole. You can get lost in these things. And he's won lots of awards.
This is cool.
I watched this because this is my favorite thing to play on the cello. And I just absolutely loved watching it. I was so happy that you had shared this, Kathy.
It's cool. It's very nice. Love it.
And one other thing, the listener pick that was a week or so ago, that You Are Not So Smart about how to talk to the vaccine hesitant. It's You're Not So Smart 213. It's almost 3 hours long, but I was on a 3-hour drive. And it is really good. There's some really good techniques and approaches and explanations of how to not alienate the vaccine hesitant, and I highly recommend it. At the beginning, they kind of go through the 10 top hit recommendations. You can just listen that far and stop if you want. But You Are Not So Smart 213. It's really worth listening to.
Rich, what do you have for us?
So this is just a wiki link to a phenomenon that interests me because, I often, what it's like when you're going to sleep and you're kind of halfway there and weird stuff happens. Okay. And you wonder, was that real? And you kind of half wake up and you go, what was going on there? And I thought, somebody's probably thought about this. This is probably a study. This is probably a thing. So I looked it up and sure enough, it is. And it's hypnagogia, if I'm pronouncing that correctly, or hypnagogia, whatever, is the experience. And I've got a wiki link in here for it. The experience of the transitional state from wakefulness to sleep. Let me see. Includes threshold consciousness phase includes hypnagogic hallucinations, lucid thought, lucid dreaming, and sleep paralysis. Ooh, sleep paralysis. I think I know what that is. It's where you're supposed to be running after something in your dream, but you can't move.
Yeah.
And then they point out that hypnagogia, actually, there's an equivalent phenomenon for when you're waking up. Okay. And hypnagogia is used to describe both, but there is a specific term for the going to sleep bit too, which is hypnopumpia. Okay. So I just thought this was amusing, that yes, indeed, it is a thing. I'm not alone. And people study this. and you can read about this and relate to it.
You know, I use the weirdness as a sign that I've kind of been sleeping, right? Because I wake up a lot. And sometimes I think I've been awake all night, but then I say, no, there was this weird period when nothing made sense. I must have been a little bit asleep, right? Yeah. Hypnagogue. I didn't know it was the thing. That's so cool.
It's a thing.
All right. Well, my pick is Landsat 9. So Landsat is a satellite that we just put up into space, and it's going to take pictures of the Earth. It's the 9th in the series of satellites. This is like a 50-year-old program or something to record the Earth's surface. And I think this is really cool. You can go, I've got Landsat 9, the NASA website. It's called Landsat Science, and you can go through all the missions and see what they did. And each one gets better. They take amazing pictures. You know, if you've seen pictures of Earth, they're just so cool. That's probably from this satellite. Just went up a couple of days ago. It will be used to measure changes on the surface, where we can separate human and natural causes of change. Look at land use, resource availability. So the data helps people make decisions. Look at deforestation, crop condition reports. The science quality of the Landsat archive, including careful calibration, allows it to serve as a gold standard for studies harmonizing multiple sources of satellite imagery. So, and this one, if I remember, the reason I found this is that I follow NASA on Instagram, and they always tell you when things get fired up into space, and they have a really nice description of what's going on. And this one apparently is polar, and it's in a low Earth orbit. It is solar synchronous, so it's synchronized with the sun. Apparently, its orbit. So it's low, so it can take nice pictures. You can find out what rocket it went up on and all this stuff. So they're still cranking out satellites, and at the same time that this went up, in the same Instagram post, they also sent up satellites for University of Colorado and Boston University. I guess they have projects, and they're in the satellite going up with them too. So things are happening. It's very cool. So you can find out all about this satellite, what it looks like, how it was put up there, what it's going to do, and so forth. It's great. I think this is really good that we need to, find out what's going on with the Earth, and this is a good way to do it. We have the tech, and every satellite gets better than the previous one. They have better sensors and better cameras and so forth. So pictures are amazing. We have 2 listener picks. Lori writes, when I see a new TWIV on my podcast player, my heart leaps with a little yippee. I'm so appreciative for all you do. That's cool. I like that. I don't know if someone has picked this, as it's been out a while. Project Hail Mary by Andy Weir. Oh, I heard about this months ago. I haven't read it yet. I heard it's great.
I know. I've been so wanting to read it.
Science fiction is not my preferred genre, but I find Andy Weir's science fiction to be more plausible than most, so I like it. The hero is a PhD scorned by academia junior high science teacher. There's an alien. There's an organism, astrophage, threatening life throughout the universe. It's great. The plot is not perfect, but there is some good science and engineering, and it made me both laugh and cry. Apparently, they're making it into a movie starring Ryan Gosling. Date not yet announced for release. Thanks again, Lori in Spokane, Washington. Yeah, I've heard it. I haven't read it either. My office is full of Dixon's books. And up on the top shelf, I saw one. It's something about the philosophy of biology by Werner Maas, a famous biologist, right? And I got it. I got on a ladder and I pulled it out. Brand new. Dixon never read it. And I started looking through it. It looks great. I don't have time to read it, but I'd like to read it. And right next to it is The Ecology of Coprophagic Beetles. Another thick book, unread. That could be interesting also. As for sure, Project Hail Mary. Has anyone read it besides?
I have not read it. I need to put this in my queue. He's written another book too called Artemis.
Yes.
Artemis too. So he's got 3 books. And I've got Artemis. I'm on a wait list for that for the library.
I liked The Martian more than I liked Artemis. And I have had many people tell me how much I would like Hail Mary Project and just have not yet read it.
All right.
Wow, how cool. He just wrote and now he's a famous author, right?
Yeah.
And the first book he was given away until someone said, you ought to sell it. And he made a fortune. Oh my gosh, they made a movie, right? And we have one from Kevin. Hello, TWIV team. I am writing from the heartland of Wisconsin, where it's a perfect 73 degrees Fahrenheit. Due to this summer's drought, particularly in the northern part of the state, some of the trees are already changing to some wonderful fall colors. This week, I came across the website oaksavannas.org. It is a glorious website that has exhaustive and quite comprehensible content for the novice ecologist. You guessed it, oak savannas. Oak savannas are one of the most decimated ecosystems in the world, with many estimates stating that less than 0.02% of the original pre-settlement oak savannas exist today. The linked website has pages describing oak savannas and the flora and fauna found there, how to restore and manage an oak savanna from start to finish, and a bit of encouragement on why we should care. Looking at the pictures and setting out to find one last weekend, I think they are just beautiful and largely unappreciated landscapes. In the spirit of microbiology, the site also happened to have been authored by the late Tom Brock and his wife, Kathy. In addition to his work in Yellowstone, most notably discovering Thermus aquaticus, Brock was an avid conservationist. They spent much of their retirement restoring the oak savanna landscape at the Pleasant Valley Conservancy in Wisconsin, which is now a public state natural area. Cheers, Kevin. This is cool. I didn't know anything about this.
The 3 major areas of oak savanna in North America are California and Oregon on the West Coast, Arizona, New Mexico, and Mexico in the Southwest, and the Midwestern United States.
I was thinking about that because I was, in the description, I was saying, holy cow, I think I grew up in an oak savanna.
Mm-hmm.
We're in counties like that. Those rolling brown hills, all covered with these low oaks.
Yeah, that's it. At least in the pictures, that's what it looks like. Yeah. Have you been to the one in Wisconsin, Kathy?
No, I haven't.
I don't know where it is exactly with respect to Madison. But if I'm ever back, I'd love to check that out. Sounds great. All right. Thank you, Lori and Kevin. That'll do it for TWIV 810. Show notes at microbe.tv/TWIV. You can send your questions and comments to twiv@microbe.tv. If you'd like to support us, go over to microbe.tv/contribute. Kathy Spindler's at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun.
I have been following that sidewalk artist.
David Zinn.
Every time I see his art pops up, I think of Ann Arbor.
Yep.
Ann Arbor, Michigan. Rich Condit is an emeritus professor at University of Florida, Gainesville. He's currently in Austin, Texas. Thank you, Rich.
Sure enough. Always a good time.
Brianne Barker is at Drew University, BioProf Barker on Twitter. Thanks, Brianne.
Thanks. It was great to be here.
I am Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWIV and Ronald Jenkins for the music. You've been listening to This Week in Virology. Thanks for joining us. We'll be back soon. Another TWIV is viral.